Okay, so we took a left turn, as I said, and we went from just talking about bacteria and resistance, and we talked about all sorts of horrifying diseases that could kill you from a simple cut or from like getting pulled under a train or getting pneumonia or something. You know, these resistant organisms and how resistance happens. But we kept saying while we were talking about that, well, you know, there's something different in their DNA. There's something different in their DNA. There's something different in their DNA. So to answer, you know, because we also want to know, well, how do antibiotics work against them? How do antibiotics kill them? So how could they possibly survive? To answer those questions, we have to go down a layer. And so the big questions you've got here go down that layer. So we're going to talk in more depth about DNA. And the first question I have for you is, what is DNA actually? So, you know, we've got this question out there, what is DNA actually? How does it get copied? Because we kept saying, well, there can be a mistake in copying, that's a mutation, and that can lead to a bacteria being different. But how does it actually get copied? Isn't there some sort of like error check? Don't you check for mistakes when you copy something? So how can there be mistakes? What does DNA actually do? So we've got this thing, we're going to figure out what it actually is structurally, but then, you know, does it just sit there? So, you know, we keep saying, you know, it's a blueprint, Whee, there's a chemical. You know, it's a chemical. It's just water, folks. Don't worry. You know, it's a chemical. Whee! Sorry. Whee! It's just water. Now you're awake. Sorry. So, okay, it's a chemical. Water's a chemical. How can a chemical be a blueprint? How can a chemical be a set of instructions? Like, okay, wait, no. I understand how, like, this is a set of instructions. There are words here that I could follow to do something.
DNA is the words.
Well, it's a chemical. It's like water or sugar or caffeine. Those are important. We'll talk about those in a little bit. But how can a chemical be instructions? So then, if it's actually instructions, what is it that actually gets built based on those instructions?
Your body.
So okay, but how does a chemical, like, okay, the water droplet that's still on your desk from when your teacher assaulted you with distilled water. So, you know, there's a drop of water. How can that translate into Devin has freckles? How can that translate into Moser has crazy curly hair?
Genetics.
Like, well, but that's a catch-all. What does that mean?
Traits.
You're just throwing out
Offspring.
stuff. Yeah, but how does that happen? Like, what actually gets built? Devin's freckles? Moser's hair? That plant, you know, why does that plant have burgundy leaves versus that plant having green leaves? How can that be based on a chemical? It's not logical. It doesn't make sense.
God.
Well, now when we talk about the nature of science, we talk about the fact that within science we can only rely on natural explanations. So God, angels, miracles, any of that is supernatural by definition. So it's not a way that we can answer questions in science. It's a whole different way of thinking. So how does this chemical make a blueprint?
I have no clue.
For other, I know. Does this not bother you?
No, I'm waiting to
This
be taught.
bothers me. So what? Now, you know, here's the other question. What does DNA and what you make from it have to do in any way, shape, or form with antibiotic resistance? So we've got this chemical. We've said that, you know, changes in the DNA. Blah blah. How does it fit together? I want this to bother you. I'm really hoping that this bothers you. I'll put a few pictures before we start this. Let me put a few pictures of other molecules up here. So my favorite molecule. Nope. Caffeine. My absolute favorite molecule. It's a bunch of carbon atoms hooked together with some oxygen, with some hydrogens, some nitrogens. does wonderful things, allows me to actually function as a human being before 9 o'clock in the morning, which is a small miracle. That's a great molecule. Here's another molecule I'm kind of fond of. This one. Glucose. Sugar. Every cell in your body, C6H12O6, is using this right now. It's a good molecule. It's a bunch of carbon atoms hooked together. Here's one that we would all die without.
Carbon dioxide.
Well, there's that one too. Water. There is the oxygen atom, or the oxygen molecule, which is 2 oxygens covalently bound. Well, that's oxygen. That's O2. So those are pretty important molecules. But the molecule that we're going to talk about today is DNA, and it's long and skinny. So a lot of these other ones are like chains and loops and circles. DNA is a little different than that, in that it is long and skinny, and it is only made up of 5 moving parts: cytosine and guanine. Okay? DNA is just this long, stringy molecule, and it's made up of only 5 things, basically. There are these 2, these are called bases. Holy cow. Bases. All about the base. All about the base. Is. All about the bases. You could make a pretty good song out of that too.
Yeah, you can.
What did I just do? Okay, so there are 4 bases that are like keys and locks that fit across. And on each one of the DNA molecule, you find one base, and opposing it, you always find the opposite base. And the easy way to remember what matches what is that apples fall from trees, A matches C, and cars park in garages, C matches D. Okay.
Gato.
Okay. Cat means gato in Spanish. Hey, that's pretty good. There's another mnemonic. So the other part is what's called the sugar phosphate backbone.
Sugar phosphate.
Sounds like a party.
Sugar phosphate backbone. That's the sides. So DNA basically looks like a ladder that somebody twisted. And I actually have a pipe cleaner model. The sides of the ladder are the sugar phosphate backbone, and the rungs of the ladder are made up of these bases paired to other bases and connected with hydrogen bonds. You drew that. So here's the thing that you have to appreciate. And it doesn't matter if you have these in the same order I do. It doesn't matter if you have all the details. That's not the important part. The important part is that you recognize how stinking massive this thing actually is. So yeah, this is DNA, and this isn't even a fraction of how long it really is. I mean, it's really hard to overestimate the size of this molecule. So what's better? What's better is the fact somebody said, "Is this actually in my body?" Not only in every, not this big, not only in your body, but Devin, in every single cell in your body.
Whoa.
There are 46 pieces of this stuff. There are miles and miles and miles and miles and miles and miles and miles of DNA inside your body in your cells. Now, fortunately, each molecule is really, really tiny, but they are really long, stringy molecules. Huh. So what's kind of cool if we go back to this, and tomorrow we're going to replicate DNA. We're going to look at how it gets copied. It's really easy to copy, and I want you to think about this overnight. Hey, what makes it really easy to match these things up is that these bases, are you writing this down? These bases only pair in a particular way. So A can only physically pair to T.
In a what?
And C can only physically pair to G. So if we were to open one of these up, and we had the sides of our ladder here, and we had, what color? C.
So we can pair A as G.
Nope. So if we have a cytosine, what's going to pair to that?
A to T. A thymine. No.
C, cars park in garages, guanine. If we have guanine, what's going to pair to that?
Cytosine.
Cytosine. If we have adenine, what's going to pair to that? Thymine. If we have thymine, what's going to pair to that? Adenine. This is called the base pair rule. Because these pairs, and actually this was really important. Remember, well, I guess we haven't talked about it in here yet. In science, we have to be able to take in evidence, observations with our senses. We have to be able to get data about phenomena. Now, you can't see a DNA molecule with your naked eye. And in the 1950s, they still didn't know what this was shaped like. But there was one thing they did know. Every time they looked at DNA and they broke it down into its chemical pieces, however many grams of guanine there were was always, always, always equal to the number of grams of cytosine. There were always equal masses of those two. And guess what was true about adenine and thymine? Same thing. Same mass. Whatever your mass of, whoops, well darn it. Whatever your mass of thymine was, it was always the same as your mass of adenine. So they figured out this base pairing rule based on that. They couldn't see the molecule. They couldn't analyze the molecule. Okay, I'll take the DNA back.